everyone. My name is Tamika John, and actually, the work I'm presenting today is work that I conducted at NYU and not in Dr. Lindahl's lab. But I conducted this work in the lab with Dr. Dan Lickman at NYU. He's an immunologist, and for those of you who aren't familiar, immunology is a branch of medical science that studies the immune system, which is basically the collection of I should point it this way, which is basically the collection of cells that protect our body against infection. Now, there are different categories of cells within the immune system. One category of cells is known as helper T cells. Now, helper T cells, they're different from other cells within the immune system because they don't really play a role of combating the pathogens or microorganisms that affect our body. Instead, they really coordinate the immune response by directing other cells where to go, what to do, like saying this is a pathogen, you need to attack this substance versus this protein. Now, traditionally, there are two categories of helper T cell. There's Th1 and Th2 for T helper 1, T helper 2. However, recently, a third category of helper T cells has been discovered, and it's known as Th17 cells. So why did we skip from 2 to 17? Th17 cells, the hallmark protein produced by Th17 cells is known as IL-17, and this really acts as a communication factor between the immune cells to say, you go here, you do this, and really direct the immune response to infection. It's been shown that there is a positive correlation with high concentrations of Th17 cells and incidence of autoimmune diseases, which are conditions in which the body's immune system identify a protein produced by the body as being foreign, and then the immune system attacks this protein. So this is the case in conditions such as hemophilia, where the body identifies factor VIII as being foreign, and it destroys factor VIII, and that's why hemophiliacs, their blood cannot clot due to the lack of factor VIII. Now, given the correlation between Th17 cells and autoimmune conditions as well as pro-inflammatory conditions, a lot of research has been conducted into what are the requirements for Th17 cell differentiation. Previously, we used a marine model which showed that a transcription factor, RORgammaT, is required for Th17 cell differentiation in mice. However, like I said, immunology is a branch of medical science that deals with the human immune system. So the next goal, so the goal of my project was to investigate what role does RORgammaT play in human Th17 cell differentiation. So how do I go about this? The first step was to silence the expression of RORgammaT. Once I silenced the expression of RORgammaT, I looked at the levels of IL-17, which I said earlier is the hallmark protein of Th17 cells. So I'm looking to see, is there a correlation with me silencing the expression of RORgammaT and the lowering of the expression of IL-17, or not? So how do I go about silencing the expression of RORgammaT? I use shRNA, which is short hairpin RNA. For those of you who are not familiar, basically we have the gene for the short hairpin RNA, and we insert the gene for the protein that we're interested in silencing. So, like right here, we'll insert the gene for, in this case, RORgammaT, a section of RORgammaT into this construct. The construct has a U6 promoter, and it also has other parts to allow for selection. Once I insert the gene onto the construct, I then transduce, depending on what I want to do, this construct into a cell. Once inside the cell, the construct enters into the nucleus, and the construct is transcribed to produce short hairpin RNA, which is actually, I don't know if you can see this very well, in the structure of a short hairpin, where there's the sense and anti-sense regions of the genes, they're bound to each other, and then there's a loop at the end. The short hairpin RNA then leaves the nucleus and then is processed by a protein called dicer into siRNA, or small interfering RNA. The small interfering RNA then joins this complex to form RISC, which is RNA-induced silencing complex. So what happens is the green part is the sense strand of the gene that we're trying to silence, and the yellow part is the anti-sense strand. So the sense strand is then removed, leaving just the anti-sense strand. The anti-sense strand then binds to the mRNA for the gene we're trying to silence, and this binding occurs because, like I said, it's an anti-sense strand. It's a complement of the gene for the protein. And then this mRNA is subsequently degraded. So that prevents, since the mRNA for this gene is degraded, it prevents translation, so there is less of this protein being produced. So to do this project, I use an mRNA for RRC, which is actually the gene for RORgammaT. We received, I believe it was 7 different shRNAs. 2 were made in-house, 3 were made in-house, and 4 were received from the Broad Institute in Massachusetts. As you can see, I'll go back. As you can see, some of these shRNAs are actually for RRC. Most of them are in the open reading frame for RRC. Some of them are outside of the open reading frame, and these two serve as controls. Because they're outside of the open reading frame for RRC, they shouldn't silence the expression of RORgammaT, which is a protein made from RRC. So before I could actually insert these shRNAs into human primary T cells, I tested the efficiency of these shRNAs because they haven't been used before. So I inserted these shRNAs along with exogenous RORgammaT into human embryonic kidney cells. We use this cell line because it doesn't produce its own RORgammaT. I inserted both of them into these cells, and then I lysed the cells and performed a Western blot. And I don't know how well you can see this, but the results show, so the top panel is Western blot, which is a way of just expressing protein levels for RORgammaT, while the bottom serves as a positive control. So you can see the levels on the bottom are relatively similar, saying that all my cells had about the same amount of protein. The ones highlighted in blue, those are the positive controls I told you about earlier. And the ones highlighted in green, those are the 2 shRNAs that showed actually knockdown for RORgammaT. So all the shRNAs were successful in lowering the expression of RORgammaT. But shRNA 7 and 8, which are these 2 highlighted in green, you can see from the top panel that there's less RORgammaT being expressed in those cells. So we took these 2 shRNAs and we produced lentiviral vectors, which are basically, they're just a method or a vehicle used to insert these genes into human primary T cells. Once we inserted these genes into human primary T cells, we ran 2 tests. The first test was real-time PCR for the expression of RORgammaT. Again, we're just looking to see that these shRNAs are effective. As you can see, compared to the control, you can see that the shRNAs were able to knock down the expression of RRC between 50% for shRNA1 and about 90% for shRNA2. We then measured for the expression of IL-17, like I said earlier, that's the main protein product of these Th17 cells. Measured for expression of IL-17, we see that compared to the controls, and the bottom switch serves as a positive control again. Compared to the controls, this is a FACS analysis. If you're not familiar with FACS, basically we do a stain for IL-17 production, and then this machine just sorts the cells and counts how many cells are expressing IL-17 versus not. So the x-axis is actually for expression of IL-17. So we say that the cells are IL-17 positive if they're on the far side of the x-axis. So as you can see, compared to the controls, when you're using shRNA1 and shRNA2, there's a downregulation in the positive number of IL-17 expressing cells, although when you're looking at this control protein, the numbers are relatively the same. And in one case, it's even higher in shRNA1. But still, you see this downregulation of IL-17 production within these 2 experimental conditions versus the control. So in conclusion, we concluded that RORgammaT does in fact play a role in the differentiation of human Th17 cells. And since this work has been done, it was a little while ago, other research is currently going on in this field to show what other proteins or what other transcription, what else is needed basically for Th17 cell differentiation, since it's such a big category because it's found to be involved in so many different conditions. So I would like to thank Dr. Nicola Manel. He is a postdoc that I worked with, Dr. Dan Lippman, NYU SIRP program, HHMI Scholars Program, and the Mark U Star program at UMBC. I'd also like to thank you for being here today to listen to my presentation. Are there any questions?
Can you use your lentivirus with this short hairpin RNA to see if you could do some sort of beneficial therapeutic action in a mouse model of some autoimmune disease?
So the thing about it is that since RORgammaT, Th17 cells, are you asking just for a method or this specific shRNA?
I guess that specific one. I mean, you sort of said that these were bad helper cells that were.
So a lot of cases of autoimmunity, although you're looking at, I don't think it's as easy as putting directly into the mouse, because if the mouse is already experiencing autoimmunity, it already has the deleterious effects of not having, these Th17 cells from its birth. So to see if there would be an alleviation, that might be kind of tricky. I think the point is to get a clear understanding of what exactly is our RORgammaT doing, what exactly are Th17 cells doing, because this is part of the immune system for a reason. They do have positive effects. Th17 cells do have positive effects. It just also has negative effects. So kind of knocking down the expression of Th17 within a disease model, that might be the best way to go, because then you'll mess up the immune system in other ways that won't be beneficial at all. So although you may or may not be able to stop this autoimmune condition, you might also mess up in ways that are more hazardous than just RORgammaT and Th17.